My name is Sally Ann Lynch. I'm a consultant in clinical genetics, and today I'm going to discuss what a gene is. First of all, if you think of yourself as a human being, you're made up of millions of cells, and inside every cell we have a nucleus. The nucleus carries 46 chromosomes, and it's these chromosomes that carry our genes. If you look at all the DNA on a chromosome, only 1 to 2% of the DNA actually makes up genes, and in total a human being has around 22,000 genes. Genes essentially are stretches of DNA that get activated and produce a protein, and that protein has a job to do wherever it is in the body. We have genes for every single part of the human body, and our genes are working on a daily basis digesting our dinner. A gene is made up of a very specific stretch of DNA, and sometimes alterations can occur in the DNA. Some alterations can cause the gene not to work and the protein becomes buckled and doesn't do its job. This can therefore cause a genetic condition. The difficulty for us in the clinical team is that all of us as humans have millions of variations in our DNA, and most of these variations are benign. It's only a small number that can cause a problem. One of the issues we have in the clinical department is trying to work out, when we see a variation in a gene, is whether this variation is causing the patient's disease or whether it is a benign finding. This is quite complex work, and internationally it's causing problems for all of us who work in the field as we learn more and more about the amount of normal human variation each of us have. It is thought that each of us have about 4 million variants in all of our stretches of DNA and about 18,000 variants in the active genetic part of our DNA. One of our jobs we do in clinical genetics when we identify a genetic disease within an individual is to advise the immediate family, but also if there are at-risk relatives, we do ask the individual patient who's affected to let their wider family know that a genetic condition has been diagnosed and that they are at risk. Our job in clinical genetics is to offer appointments to those at-risk relatives, but it is up to the individual who is affected to contact their relatives directly. We do not contact at-risk relatives directly ourselves. Then when we see the at-risk relatives, we can arrange for genetic testing to see if they've inherited the gene or not themselves, and to see if their children are at risk or not. If somebody does not carry the affected gene, then their children are not at risk.